Hello everyone and welcome to video tutorial 21. You should now know all about the two types of variation and you should be able to pause the video and answer the questions. So question 1 is which line in the table below identifies the type of variation shown by each of these human characteristics? Question 2 is describe what the term discrete variation means. And question 3 is when an individual's phenotype is controlled by only one gene, name the type of inheritance. So pause the video and try to answer this before I go over the answers. So the answers are that we know we can measure height, so height's continuous. We know we can measure weight, so weight's continuous. And we know that earlobes are either attached or not attached, so it's a yes or a no, so they are discrete because it comes in categories. So well done if you managed to get A for that. Then it says describe what the term discrete variation means. So you should have put that it's where the variation shows clear-cut differences and they can be placed into distinct groups or categories. And then the third question is when an individual's phenotype is controlled by only one gene, name the type of inheritance. So it would be single gene inheritance. So well done if you managed to get that correct. If you didn't manage to get that correct, then you need to go watch video tutorial 20 again. So today we're going to continue to learn about variation and inheritance, which is still part of key area 4. If you want the worksheets that go with the video, remember to pause the video and go to 2 multicellular, 4 variation and inheritance, 2 consumables and activities, and the PowerPoint is found in 1 PowerPoint and is called 4.2 variation and inheritance. Our next learning intentions are to be able to explain the genetic terms, which include gene, allele, phenotype, genotype, dominant, recessive, homozygous, heterozygous, P1, F1 and F2. Be able to carry out monohybrid crosses from parents through to F2 generation and to know the reason why predicted ratios are not always achieved. To be successful, you will need to be able to write down and explain the definition for all genetic terms, successfully complete Punnett squares of monohybrid crosses, be able to predict the phenotype and genotype from monohybrid crosses and explain why predicted and observed ratios are not always achieved, and describe how you are more likely to get a predicted ratio. We all have different features like hair color, eye color, and these things are described as our characteristics and they all vary. But how do we get these characteristics? These characteristics are determined by genetic information, and this information is passed from one generation to the next. So each individual receives 2 pieces of genetic information for each characteristic. One piece of the genetic information comes from their mum, and one piece of the genetic information comes from their dad. These can be the same or different pieces of information, and sexual reproduction involves combining genes from separate parents and so produces genetically distinct offspring, which leads to variation. So what is inheritance? So all organisms receive genetic information from both their parents, therefore they will have some characteristics passed on to them. Inherited characteristics can appear generation after generation. This can be shown in a family tree. You should have looked at the Simpsons family tree in 3rd year, and we will look at family trees in more detail later on in the video. An example of an inherited characteristic that was passed on from generation to generation is the Habsburg lip. The Habsburg lip is a disfiguring genetic disorder where the lower jaw outgrows the upper jaw. Many members of the European royal families in the 16th and 17th centuries inherited the Habsburg lip, and several artists of this time captured this royal family feature. The Habsburg lip could be passed on to a child even if only one parent had it, so it was dominant. So there is a picture of the Habsburg lip. So even if one parent had it, then a child would have it. Another example of an inherited characteristic is night blindness. This is the inability to see clearly in the dark, and this image shows how a person with night blindness would view things at night. So you need to know the word genotype. So the genotype is the genes that an organism has. It's the genetic makeup of the alleles an individual has. So basically all the organism's genes are its genotype. So you need to write this down over and over again and memorize it. Another word you need to know the definition of is phenotype. So The phenotype is the physical characteristics possessed by an individual, so basically what an organism looks like. So this might be coat colour in guinea pigs, leaf shape in tomato plants, wing type on fruit flies, etc. So again, you need to write this down many times and memorise the word phenotype before you move on. As I have already said, the genotype determines our phenotype. So our genetic information determines our characteristics. Usually you would make a note on this and stick in the diagram, which you can get from the consumable section. So we need to know how to be able to do genetic crosses and how to be able to read them. So firstly, we need to know what these letters represent. So what does the P, the F1 and F2 represent? So you've got asked the question which letter represents parents, which letter represents first generation, and which letter represents the second generation. So you can pause the video and try to answer this before I go over the answers, because it's pretty straightforward and you probably will be able to guess it. So which letter represents the parents? This would be P to represent the parents. And then which letter represents the first generation? It's going to be F1, which represents the first generation. And then which letter represents the second generation? It would be F2, represents the second generation. So well done if you managed to get that correct. Now we're going to look at dominant and recessive. So you need to try to answer these questions using the cross in this corner that's just popped up. So try to answer the questions, pause the video before I go over the answers. So the answers are: the parents are black and white, as you can see in the genetic cross. The first generation, which are F1, are all black. Then we have F2 generation, which are the second generation, which are black and white. So in F1 there are no white guinea pigs. In F1 they are all black. In F2 both phenotypes are present, so there's black and white. In F2 black occurs in the greatest numbers, and the phenotype ratio of F2 would be that you have 1, 2, 3 black to 1 white. So dominance means that there's more of something, so black is dominant and white is recessive because there's less of it. So let's look at the terms dominant and recessive in more detail. So well done if you managed to get any of them correct. So now you would make a note on dominant and recessive, and you need to know the difference between each. If it's dominant, the gene will always be expressed. You only need one copy of the gene for its phenotype to show. It is shown as a capital letter, so for example, capital R. Whereas recessive, the gene can be hidden. You need 2 copies of the gene for the characteristic to be expressed. And the recessive genes are shown by small letters, so for example, little r. So for this diagram below, you've got a dominant brown, so it could be big R, and it doesn't say that it's hidden. And then for your recessive blue, it would be little r, and then for your child, it would be big R, little r, because the blue gene is hidden, so it would still display the brown phenotype. So make sure you go over and over this and learn the difference between dominant and recessive before you go on. So now let's look at true breeding. So use this genetic cross and answer the question. So how does the colour of the F2 offspring compare with the colour of the parent? So the parents in both crosses are said to be true breeding. This means that the offspring of true breeding black guinea pigs are always, so what do all of these look like? What colour are they all? Okay, you're right if you said black. And the offspring of true breeding white guinea pigs are always, well, let's look at all of these. They're all white, so well done if you managed to say white. So the parents in both crosses are said to be true breeding. This means that the offspring of true breeding black guinea pigs are always black, and the offspring of true breeding white guinea pigs are always white. So well done if you managed to get that correct. So true breeding is when offsprings and parents always show the same characteristic. So you'd want to make sure you had a note on this. So make sure you learn that true breeding is when offspring and parents always show the same characteristic. You need to make sure you make a note and learn the definition of the word allele. Alleles are different forms of the same gene. So, for example, the gene of hair color can be red, it can be blonde, it can be black. Okay, so common question is, what are the different forms of a gene? And your answer to that would be allele. So make sure you learn this definition. It comes up over and over again. Every normal body cell in an organism carries two matching sets of chromosomes, one originating from each parent. Thus, every body cell has two alleles of each gene. Alleles are located at the same distance of a chromosome. Alleles can be different on each chromosome or they can be the same on each chromosome. So we've got individual A and we've got two different alleles for this one. So one for blue eyes and one for brown eyes. The person will have brown eyes because brown is the dominant characteristic. We've got individual B, which has got two brown alleles, so it's going to have brown eyes. And we've got individual C, which has got two alleles for blue eyes, so this person is going to have blue eyes. So make sure you understand the term allele, make sure you understand the diagram at the bottom before you move on. This is just to help you note that the alleles are on the same position on each of the chromosomes. So you've got your allele for purple flowers and your allele for white flowers. So they're right on the same position. So when sex cells, also known as gametes, which would be your sperm and your egg in your case of humans, are formed, the pair of chromosomes will be split up. So each gamete only gets one chromosome and so one copy of the allele for each of the genes. So one chromosome goes into the egg and this chromosome will go to the other egg. So this means that when one gamete fertilizes another, the resulting zygote will have two alleles, one from each parent. So the appearance of the organism depends on which allele is dominant and which allele is recessive. So let's have a look at symbols. So symbols represent the alleles. So the dominant and recessive alleles of a gene are often represented by symbols. Dominant alleles are represented by a capital letter, and recessive alleles are represented by a lowercase letter. An organism's genotype is always represented by two letters per gene, since everybody cells has two alleles for each gene from each of your different parents. One from your mum and one from your dad. Depending on whether the alleles are dominant or recessive will depend on which phenotype the offspring will receive. So for example, this person has got a capital E from their dad, which is dominant, and a small e from their mum, which is recessive. So they would share that characteristic the same as their dad. The next thing that you need to know is the difference between the terms homozygous and heterozygous. When an organism possesses two identical alleles of a gene, its genotype is said to be homozygous, and it's also said to be true breeding. So this can be, for example, capital R, or small r, because both the alleles are the same, and both the alleles are the same. It doesn't matter which letter is used because it just represents the alleles of a gene. So when an organism possesses two different alleles of a gene, its genotype is said to be heterozygous. So in this case, it would be a capital letter and a small letter, and I've just used R for an example. So capital R, small r. You can try to remember this because homo usually means if you're homosexual, you'd be attracted to the same sex. So homozygous means the same alleles. Heterozygous and heterosexual usually means that you're attracted to a different sex. So you can remember that these are two different alleles. So you've got an example at the bottom. So you've got capital S for the P shape, which is smooth. And then you've got capital S and small s for the P shape again, which is smooth. Every time there is a capital letter, it means the phenotype will be the dominant one because the recessive one is hidden. And then for the last one, you've got two little s's, which is your rough seed. So this one, number one, And number 3 seeds will be homozygous because their alleles are the same, whereas number 2 will be heterozygous because its alleles are different. So when alleles are the same, it's homozygous. When alleles are different, it's heterozygous. So make sure you know that before you move on. You should now be able to pause the video and write the description of whether each of these is heterozygous or whether they are homozygous. So pause the video before I go over the answers. So the answers are that the first one would be homozygous because the two alleles are the same, because you've got big T, big T. The second one is heterozygous because you've got big T, small t. And the bottom one is homozygous because you've got little t, little t. So well done if you managed to get that correct. If you didn't, then go back to where it says homozygous and heterozygous and learn it. So true breeding organisms are homozygous and will always pass the same alleles to their offspring. So for example, if the blacks were dominant and were big R, then all of them would be big R, because they would pass it through and all of them would be homozygous. And if the whites were recessive, then all of the mice would have the genotype little r, and again would all be homozygous. So if it's true breeding, it is homozygous. Now in class we would complete the guinea pig worksheets and the SpongeBob extra worksheets. So if you go to consumables, you can print these off before you go to the next part of the video. So in the guinea pig worksheet, you will see that your parents are homozygous. So one's homozygous dominant, so capital R, and one's homozygous recessive. Now if it's recessive, it always contains the two small lowercase letters. And then if we were going to cross both of them with each other, you'd get this R, so it'd be capital R with this R, them two alleles, so we get big R, small r. And then if you cross the other two alleles together, you'd still get a big R and a small r. So all of the offspring would then be big R, small r, so they'd all have long hair because they contain the dominant gene. So well done if you managed to get that correct. Long hair is the dominant gene. So now you should be able to pause the video and fill out the first part of the worksheet and answer the questions, which are what has happened to the little r genes and what phenotype are the guinea pigs. So pause the video and try to answer the question. The answer is that the little r gene is now masked with the dominant big R gene, and the phenotype is therefore the dominant long hair for all of the first generation guinea pigs. So staying with the guinea pig worksheet, it now says that two guinea pigs are homozygous. Mark this on your worksheet. And if you were shown a long-haired guinea pig, what two possible genotypes could you have? And how would you describe a genotype of capital R, small r? And what are the alleles for hair type in the guinea pigs? So pause the video and try to come up with the answers before I go over them. So the answers are that the two guinea pigs that are homozygous would be your two parent guinea pigs. So your parent with the long hair, which has capital R, and your parent with the short hair, which has small r, So if you were shown a long-haired guinea pig, what two possible genotypes could it have? It could either be capital R, or capital R, small r. And how would you describe a genotype of capital R, small r? It would be heterozygous. Then it says, what are the alleles for hair type in the guinea pig? So the alleles would either be capital R or small r, or your two different forms of the gene. So well done if you managed to get them correct. If not, then you need to go back and watch the video again. So in the worksheet, you're then asked to cross the F1 generation together. And we know the F1 generation were all capital R, small r. So we want to get the F2 generation, so the second generation, by crossing a big R, little r, so that's the alleles from one parent, and then the alleles from another parent are big R, little r. So we do this by setting up a little Punnett square. So from one parent we've got a big R, and from the other parent we've got a big R, so we get big R, big R. From one parent we've got a big R, from the other parent we've got a little r, so we've got big R, little r. From one parent we've got a big R, from the other parent we've got a little r, and from one parent we've got a little r, and from the other parent we've got a little r. So we have got one homozygous dominant, two heterozygous dominant, and one recessive. So if it's got a big R, remember it's got to have the dominant phenotype, so that would be long-haired guinea pig, and you'd stick your long-haired guinea pig in. Same with these two that have got the dominant letter, so long-haired and long-haired. And then we have one short-haired. So our genotypic ratio would be capital R to 2 capital R, small r, to 1 small r, small r. Now our phenotypic ratio, so that would be genotypic. Our phenotypic will be different. Sorry, it's hard to write with this pen. So our phenotypic ratio would be 3 long-haired to 1 short-haired, and you're going to learn that this is a famous 3-to-1 ratio that you're going to have to know. So you've got 3 of the dominant characteristic and 1 of the recessive characteristic. So well done if you managed to get that right without help, and you should now be able to complete that guinea pig worksheet. So Gregor Mendel discovered basic principles of heredity by carrying out various experiments on peas. So pause the video and watch the link below to look at inheritance crosses, or go back to the link at the end of the video. As has already been said, Gregor Mendel is a famous scientist, and monohybrid inheritance was first studied by him in the 19th century. He is an Austrian monk, and he only studied one clear characteristic at a time in breeding experiments, which we call crosses. The crosses are worked out using Punnett squares, which you will need to learn how to use. A monohybrid cross is used to predict the chances of offspring inheriting a genotype, so like you getting a characteristic from your parents. A monohybrid cross is when you breed or cross two individuals to find out about one trait, so for example, eye colour. So let's look at an example of this. In pea plants, red flower colour, capital R, is dominant to white flower colour, small r. If we were to cross a homozygous red plant with a white plant, we would set a cross out as follows. So first of all, we know what the words dominant and homozygous mean. So if we have a dominant homozygous plant, we will have capital R, and you should be able to tell me that before I say it. And for the recessive form, and it's homozygous as well, we should have small r, small r. Our parent phenotype, like we've already said, should be red and white. Our parent genotype, like we've just worked out, should be capital R for the alleles and small r for the white alleles. Then we should have our gametes. So for our gametes, we've got to take it from our red and white parents. So we've got capital R. So that's the two combinations we can get from the red. from the white we can get small r or small r. So our F1 genotype, if we cross the gametes together, we would get big R, we'd get big R, big R, small r, and big R, small r. So there'd be 100% big R, small r. So our F1 phenotypes are all going to be red because they all contain the capital letter and the white little letter will be hidden. So we should have 100% red. So all of the offspring are red and they are all heterozygous because they contain two different alleles. In a monohybrid cross, you would then cross the F1 generation, and we know that the F1 generation were both red and heterozygous. So if we take two of them, so F1 phenotype is going to be red and red. The genotype for this is going to be capital R small r and capital R small r. And if we cross them together, well, first of all, we need gametes, so we would have big R or small r from that red parent, and then big R or small r from this red parent. And then that would give us the combination of either big R, big R, big R, little r, big R, little r, or little r, little r. So the phenotypes would be, we need to find out which ones have capital letters. So these three have capital letters, so they're all going to be red. And then the last one, which doesn't have a capital letter, is going to be white. So this then gives us a phenotypic ratio of 3 reds to 1 white. Now this is the famous 3-to-1 ratio, so if you cross any two heterozygous things together, you will always get a 3-to-1 ratio. So you'll always get 3 of the dominant form and 1 of the recessive form. And you need to know that. So crossing two heterozygous is always going to give you a 3-to-1 ratio. So let's review what was on the last slide. So if you complete the F1 generation cross, then you get a ratio of 3 dominant to 1 recessive. So this can be split up into 1 dominant homozygous, 2 dominant heterozygous, and 1 recessive homozygous. There is an easier way to work things out that is less time consuming, and that is the method of using a Punnett square. So Punnett squares make things easier and they also make things neater. So we're going to do the exact same thing as we've just done, but we're going to use a Punnett square. So R equals red in your pea plant and a small r equals white in your pea plant. So obviously red is the dominant form and white is the recessive form. So it says if we cross a homozygous red plant with a homozygous white plant, the following Punnett square would be produced. So you've got your, try to work out your F1 generation. So we know that our parents are homozygous and red is dominant, so it's going to be capital R, and we know for the white plant it's going to be small r, because it's recessive. So here is our gametes, big R for the first parent, and then our gametes for the second parent go here, so small r, and then we would cross the big R and small r to get big R, small r. We'd cross this one and this one to get big R, small r. Then we'd do this one and this one to get your big R, small r, and then we do this one and this one to get your big R, small r. So all of them would be big R, small r, so all of them would be heterozygous, and all of them would be the dominant phenotype, which would be red. So notice if you cross two homozygous that are different, so one dominant and one recessive, you always get all offspring that are heterozygous dominant. So that's your gametes for your red parent and your gametes for your white parent. Now you don't have to do it in a table, you can just do it like that. So you just make a little square and you just put your big R, small r, and then you get your big R, small r, big R, small r, big R, small r, so you get 100% big R, small r, and 100% red. So then in our monohybrid cross, we then crossed our two F1 generations so we could find the F2 generation. So crossing the F1s would be big R, and big R, small r. So if plants are heterozygous, what would the square look like? So you can pause the video and try to come up with what the offspring will be, because there's a famous ratio that you should be remembering. So you've got big R and small r from one parent and big R, small r from the other parent. So then that will then give you big R, big R, small r, big R, small r, and little r, little r. So we have then got 3 with capital letters, and then we've got 1 with 2 small letters. So we are going to have a phenotype of 3 reds, because they've all got capital letters, to 1 white. So our famous 3:1 ratio. And then we've got 2 homozygous, so 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous. So you've got your genotype ratio of capital R to 2 capital R to 1 small r, and the phenotype would be 3 red to 1 white. Now you can say that they're 75% red and 25% white, and the way you would work that out is there is 3 out of the 4 offspring are going to be your red. So 3/4 of 100, or 3 times 100 divided by 4, is going to give you 75%. So 75% are going to be red. You can also do that with the white. So we know that 1 out of 4, so 1/4 of 100, is going to be 1 times 100 divided by 4, which is going to give you 25%. So 25% are going to be white. Sorry, that's not a very good 5. Okay. So just to review what's already been said, in a monohybrid cross, all the F1 generation will show the dominant characteristic. So for example, red, and this is because you will have a cross of your 2 homozygous parents, and if you cross 2 homozygous parents, you always end up with a heterozygous offspring. So all of the offspring will be heterozygous and will show the dominant characteristic. Now if you cross your 2 F1 generation and you get your F2 generation, you will get your 3:1 famous ratio. So 3 of the dominant form and 1 of the recessive. This is the famous 3:1 ratio, and you get it when you cross 2 heterozygous parents. So if you cross 2 homozygous parents that are dominant and recessive, you will get 100% heterozygous, capital R, small r. If you cross 2 heterozygous, you will get 3 dominant and 1 recessive. So you need to know that, you need to remember that. If you're struggling, then go back. Also, you need to know what the term carrier means. So a carrier is not someone with a disease, but can pass the disease on. So they are always heterozygous, and the disease must be recessive. So they themselves do not have the disease, but they carry it, so their child can end up with having the disease. So now let's look at some examples of Punnett squares. So 2 parents are heterozygous and can roll their tongue. What are the chances of their baby being able to roll its tongue? So we've got capital T is the allele for tongue rolling, and small t is the allele for non-tongue rolling. Now, you can use your own letters unless they tell you in the question what letters to use, and then you have to stick with whatever letters you've been told to use. Now we know that the 2 parents are heterozygous, so you should know what the offspring are already without me having to say anything. So it says that the man has a genotype capital T, small t, so 50% of his sperm will have the allele capital T, and the other 50% will have the allele small t. The woman also has a genotype capital T, small t, so 50% of her eggs will carry the allele capital T, and the other 50% will carry the allele small t. So try to put the information in a Punnett square yourself. So pause the video and try to do that before I go over the answers. So the answers would be that you would have a Punnett square and you'd set up with capital T small t from the male, capital T small t from the female, and then you would cross them. So capital T would be your answer, and then capital T small t, and then capital T small t, and then two little t's. So this gives you your famous 3:1 ratio. So you've got your 3 dominant and your 1 recessive. So 3 can roll their tongue and 1 can't roll their tongue. And then you've got your genotype ratio of 1 capital T, to capital T small t, and 1 little t little t. So you've got your 4 possible combinations in the fertilized egg. So normally you draw it like this, and these parents will then have a 25% chance of getting a baby who is capital T, and you get that by doing a quarter of 100 like I showed you before. And then it's got 50% chance of getting a baby with capital T small t because there is 2 of them. So 2 divided by the 4 times by 100 gives you your a half of 100. And then you've got your 25% chance of getting a baby with small t. So that would be a quarter of 100. So what chances does the parents have of getting a baby who can roll their tongue? It would be a 75% chance of the baby rolling its tongue. So what is the ratio of tongue rollers to non-tongue rollers? It would be the famous 3 tongue rollers to 1 non-tongue roller. Well done if you managed to get that correct at the very, very beginning. And remember that if you cross 2 heterozygous things together, you get 3 dominant and 1 recessive. So here it is highlighted for you. So this is the famous 3:1 ratio that you get when you cross 2 heterozygous parents. So try to do the next question by pausing the video and doing a Punnett square before I go over the answers with you. So the question is, a tongue rolling man who is homozygous dominant, capital T, has a baby with a woman who can't and who is homozygous recessive, which is small t. You should actually be able to work this out in your head and not have to do a Punnett square because we've already went over what happens when you cross a homozygous dominant and a homozygous recessive together. So try to write down the answer before I go and to reveal the answer to you. So the answer would be that you would draw your Punnett square with your capital T, and then your small t, and then you'd cross it so it'd be capital T small t for all of them. So the chances of them being able to roll their tongue would be 100%. So all of the children will have the genotype capital T small t, and all of them will be able to tongue roll. So 100% chance. Try this example on your own. So tongue rolling is caused by a dominant gene. A woman who can't roll gets a baby with a man who is heterozygous for the trait. What are the chances of their baby being able to tongue roll? So pause the video and try to work this out before I go over the answer. So the answer would be, first you need to work out what you are going to cross. So a woman who can't roll their tongue would be small t, and a man who is heterozygous would be big T, little t. So that's the first thing that we need to work out. Then we need to put both of these in a Punnett square. So you've got your little t crossing with your big T and little t. So you get big T, little t, big T, little t, little t, little t, little t, little t. So you have got 2 that have big T's, so 2 that are going to be able to roll their tongue and 2 that are not going to be able to roll their tongue. So you then need to work that out. So 2 divided by 4 times by 100. So 2 times 100 divided by 4 gives you 50%. So 50% chance of the child being able to roll their tongue and 50% chance of them not being able to roll their tongue. And it would be a 1 capital T to your 1 little t because it's a 2 to 2 ratio, which can be simplified. So 50% chance of the child being able to roll its tongue. So well done if you managed to get that correct. The next question that you need to work out is, having dimples is a dominant trait. A homozygous dominant male has a child with a heterozygous woman. What are the chances of their child having no dimples? So you know that a homozygous dominant, what that means and what heterozygous means, so you need to work that out, and then you need to do a Punnett square. Pause the video and try to do that now before I go over the answers. The answers are that you would have a capital D for your homozygous dominant male, and then for your heterozygous women it would be a capital D, small d. So you put that in a Punnett square, so you get capital D, then capital D, capital D, capital D, small d, capital D, small d. So you've got all of them have a capital letter, so all of them will have dimples. So it's going to be 100% chance of the child getting dimples. And then you'd have a 1D, capital D to a 1 capital D, small d, because it would be a 2 to 2 ratio, which would be simplified to 1 to 1. So well done if you managed to get that correct. So all of them will have them. Tracking inheritance can be fun to see who can roll their tongue or have dimples, but it can also have serious consequences as some conditions are genetic. So common genetic conditions include cystic fibrosis and sickle cell anemia. Doctors can use crosses to see how likely it is that your child will have these genetic conditions. So the next question is based on this. Linda is a cystic fibrosis carrier. This means that she does not have the condition but carries the allele. She is heterozygous. She falls in love with David, who is also a carrier. What are the chances of them having a child with cystic fibrosis? So try to answer the question yourself, pause the video and make your Punnett square, etc. and before I go over the answer. So the answer is, firstly, we need to work out the genotype of the parents. So we know that Linda is a carrier, so we're going to use N, so capital N and small n. And we also know that David is a carrier, so he's also going to be capital N, small n. So you should be able to work out what the offspring are going to be without the Punnett square. But let's do the Punnett square anyway, just to check. So we should have a capital N, capital N, capital N, small n, a capital N, small n, and a small n, small n. So we know that this person and this person would be carriers. This person would be normal and wouldn't have the disease at all and wouldn't be a carrier. And this person would have the disease. So what chance of them having a child with cystic fibrosis? So that would be 1 out of 4, which is 25% chance of them having a child with cystic fibrosis, 50% that they'd have a carrier, and 25% that they'd have a totally normal child. And we should have been able to work that out relatively quickly because we always know that we should be getting a 3 to 1 ratio when we cross two heterozygous things together. So well done if you managed to get that correct. If not, then you might want to go to work through the examples again. If something has a recessive phenotype, we know that the alleles are always going to be 2 little letters. But if something has a dominant phenotype, how do we know what alleles they are? Because it could be heterozygous, like big R, little r, or it could be dominant, like big R, big R. So we can find this out by doing a test back cross. So this means crossing the dominant individual, who will either be homozygous, capital R, or heterozygous, big R, little r, with a recessive individual. So let's go through how you would do a back cross test. If you cross a heterozygous with a recessive, your gametes would obviously be big R, little r, and then 2 little r's for your white plant. And if you put it into a Punnett square, you would get big R, little r, big R, little r, and then little r, little r, and little r, little r. So you get 50% are going to be the dominant form, so red, and then 50% are going to be the recessive form, so white in this case. So no matter what you do, if you cross your heterozygous with your recessive, you are always going to get 50% of the dominant and 50% of the recessive. Whereas if you cross your homozygous dominant big R with your homozygous recessive little r, your gametes are going to be big R, and little r for white. if you put them in a Punnett square, you're going to get 100% big R, little r, so they're all going to be red. So that is how you can tell the difference. So if it's heterozygous, it's going to be 50-50%, and if it's homozygous, they're all going to be red. So if all of the babies have the dominant phenotype, the parent must be homozygous dominant. So for example, capital R, capital R. If 50% of the babies have the dominant phenotype and 50% have the recessive, the parent must be heterozygous dominant. So big R, small r. So if you don't understand this, go back and watch it again. Another thing that you might frequently get asked about is the observed versus predicted phenotypic ratios. So there is often a difference between the observed and predicted figures. Why does this happen? And your answer needs to be that fertilization is a random process and it involves the element of chance. So, for example, you might get asked something along the lines of, the child was predicted to have curly hair. Why did the child not have curly hair? Your answer would be because fertilization is a random process and it involves the element of chance. The next question that frequently gets asked is, how could you improve the chances of getting a ratio such as the 3-to-1 ratio? And the answer for this always needs to be to increase the sample size. So two things that you need to know are, why is there a difference between the observed and predicted figure, and you will answer that fertilization is a random process and involves the element of chance. And the next thing is, how could you improve the chances of getting a 3-to-1 ratio? That would be increase sample size, so having more children. So make sure you know how to answer them both questions, write them out over and over again, say them over and over again. Now you should be able to work through the consumable called the superhero worksheets. It's called 13, which is in the consumables, and that'll help you to practice your Punnett squares. You can also do activities 11.1 and 11.2, which are also found in the consumables. Let's now look at family trees. So you should have looked at family trees in third year. You should have looked at this one with the Simpsons family tree, and you should have got asked a bunch of questions about it. So you should know that Abraham is Homer's dad, Mona is Homer's mum, Herb is Homer's brother, Homer has 3 children, Bart is his son, Lisa is his daughter, and Maggie is his daughter. He is married to Marge. Marge's parents are Clancy and Jackie. She has 2 sisters, which are Patty and Selima has a child called Ling. So let's go into family trees into more detail in a second. Now pause the video and go to the link and watch about family trees, or you can come back to the video at the end. So family trees are diagrams that can be used to show the transmission of alleles over a number of generations. The following family trees show how the ability to roll for the tongue allele is transmitted through 3 generations. So a pedigree chart is a type of family tree diagram. Pedigree charts are used to show the inheritance of a particular characteristic through generations of the same family. Males are represented by symbols, for example squares, and females are represented by a symbol, e.g. circle. These can be coloured or blank depending on whether or not you have the characteristic. So for example, we have this tongue rolling one. So if you're male, you'll be square. If you're female, you will be circle. If you have your ability to roll your tongue, you will be coloured in black. If you have the inability to roll your tongue, you will be coloured in blue. So look at this family tree. Who is Jack's grandparents? So pause the video and try to write out Jack's grandparents before I go over the answer. So the answer would be Alan and Betty are Jack's grandparents, because Jack is down here. Anne and Tim would be the parents, and then Alan and Betty the grandparents. So well done if you managed to get that correct. So then it says, look at this family tree. How many siblings does Sam have? So again, pause the video and try to answer it before I go over the answer. So the answer would be, first of all, we need to find Sam, and then we need to look at the siblings. So that would be all of the people that are on the same branch. So Ben, Jan, Jill, Tom, and Anne. So 5 siblings. Right, the next question is, who is Anne married to? So pause the video and try to come up with the answer on your own. So the answer would be, first of all, you need to find Anne. And when there is a branch coming out of it, that means that the person is married to that other person. So in this case, it would be Tim. The next question is, look at the family tree. How many brothers does Jane have? So again, pause the video and try to come up with the answer before I go over it. So the answer would be, first of all, we need to find Jane. And then we need to look for a square because we know that the squares are for the males. So the answer to this would be 1. So look at this family tree. How many sisters does Jill have? Pause the video and try to answer the question before I tell you the answer. So the answer would be to find Jill, first of all. And we need to look at for sisters. So we know sisters and females are circles. So it would be 2 in this case. And it says, how many children does Sue have? So again, pause the video and try to answer the question before I go over the answers. So the answer would be, here is Sue, and when it goes down a stage, that's going to be the person's children. So 1, 2, 3, 4. Sue has 4 children. So well done if you managed to get all that correct. If you didn't, then go back to the start of the family tree and try to work through who is married to who. So obviously we have the grandparents up here, we then have their children, and then we have whoever they're married to on the side branch, and then we have their children, and then it would go down further to their children if they had any, and so forth, and it would just continue like that. Family trees can be used to identify the phenotypes and genotypes of individuals. So the question is, if tongue rolling is dominant, what genotype would a non-roller have if we use the letters capital R and small r? So we know that the dominant form of the gene is always going to be the capital letters, and we know that the recessive form of the gene, which is your non-roller, would have to be 2 small r's. So all of the ones that are recessive have to have 2 small r's. So let's try to work out the genotype of this family tree over here. So we already know the phenotypes because we've got a key for that. So if it's female, it's going to be a circle. If it's male, it's going to be a square. If they can roll their tongue, they're going to be white. If they can't roll their tongue, they're going to be black. And we know that the blacks are now going to be small r, because it's the recessive form of the gene. So first of all, you'd always put the recessive form of the gene in first. So all of the people that are black would have the genotype small r, small r. We then need to work out what all of our white ones are going to be. So we know that this has to have a big R of some sort. This has to have a big R of some sort. Now, because all of the offspring of these 2 people are not all big R, small r, we know that it has to be a big R, small r parent to form this. Because if you crossed your big R, and big R, and small r, you'd get all big R, small r. So all of your offspring would therefore have to be white, and that is not the case. Some of them are black. So it has to be 50-50. So it has to be heterozygous. So let's look at the other side. The other side is the same. Some of the offspring are white and some are black. So this is going to have to be big R, small r. So this one will be big R, small r. This one will be big R, small r. So when we cross our big R, small r, with our little r, we should get 50-50, and we should know that from the test back across. So big R, small r, and then little r, little r, little r, little r. So you get 50% big R, little r, and 50% little r, little r. So these two then also need to be big R, small r, small r, and we've worked out the genotypes of all of them. So the key is to firstly put in all of the recessive ones, and then is to look at what type of offspring that you get. Okay, let's try some examples of this. So pause the video and see if you can try to work out the genotypes of all of these people in this tree. You should be able to print out the tree, it should be in consumables. It tells you at the top that capital R is normal and little r is sickle cell anemia. We should be able to work out what ones are dominant and what ones are recessive. And the reason being is you have all of these are black, and you've crossed your two parents together, so obviously this white one is hidden, so they must have a dominant gene of some sort. So you know that all of your black ones must contain a capital R, and you must know now that all of the ones that are white are going to be recessive. So Alan will be recessive, Tim will be recessive, Mary will be recessive. Now you need to work out what Betty is. Now if there's 100% dominance, then Betty has to be big R, big R. Okay, and if you cross big R with little r, all of these are going to be big R, little r. Now that means if you cross big R and little r, you get 50% big R, little r, and 50% little r. So Jack's going to be big R, little r, and Mary will be little r, little r. Right, so now we just need to work out Sue and Sam's children. So Sue, if she was big R, then it would not be possible to get Jane, because big R times big R, little r will give you big R, 50% of big R, and 50% of big R, little r, none of which are the recessive form, and Jane's the recessive form. So Sue must also be big R, little r. Now if you cross two heterozygous together, we should be getting our 3:1 famous ratio. So Bill, Lynn, and Mel are either going to be big R, or big R, little r. And it's not possible to tell which one they are. You could only tell that if you crossed them with a homozygous recessive. So well done if you managed to get that without any help. If not, then watch over how we did it again. So remember, you find all the recessive and then you work your way back. Now try to work out the genotypes for this family tree on your own. So pause the video and try to work out all their genotypes before I go over the answers. So the answers would be, firstly, ability to roll your tongue is dominant, and you can tell that because when Granny and Grandad are crossed and they've got the two different phenotypes, your aunt and your dad have the ability to roll the tongue, which means that they're dominant. So that means that all of the people with the inability to roll their tongue must be recessive. So they would be little r, little r. So all the people on this side are going to be little r, little r, little r, little r, little r, little r. Now, because there's 100% big R, small r down here, then granny must be big R because it's not 50-50. And then if you crossed your big R and your little r, you would get big R, little r, big R, little r, big R, little r, big R, little r. You can put these all in Punnett squares if you want to, but you should know that if you cross two homozygous, one dominant and one recessive, you will get all heterozygous big R, small r. Now, if you cross big R, small r with little r, you should get 50% big R, little r, and 50% little r. Again, you could do this in a Punnett square if you wanted to check. Okay, so well done if you managed to get that correct. If not, then you need to watch the video on the trees again until you understand it, or you need to come get help from one of your teachers. So since each organism receives two pieces of information for this characteristic, we need to work out the genotypes, which we have just. So well done if you managed to do that without my help. So a family tree might be used by a genetic counsellor when advice is needed by a couple concerned about passing on a genetic disorder to their child. So for example, cystic fibrosis. This would only be done if the disorder is known to exist in their families. So cystic fibrosis is a genetic disorder and it's caused by the mutation of one gene. And it causes the sufferer to produce mucus that is thick and stickier than normal, which leads to congestion of the lungs and other organs. If the condition is known to exist in families, couples may want to seek advice about the risk of their children inheriting the disorder, and that's called genetic counselling. So there's an animation about this, which you can pause the video and go to now, or you can come back to it and go to it at the end. So what are the chances of Mary and Jack's child having cystic fibrosis? So we have our key and we've got our family tree. So you try to work it out before I go over the answers. So the answers would be that we know that the male non-sufferer is yellow and the red is the sufferers. And we know that the yellow here must be dominant because if you cross these two, the red is masked. So we can then put for all of the ones that are recessive, the letters little r, little r, little r, little r, little r, little r, or you can use whatever letters that you want. And then we need to work out what these yellows are. So we have 50-50, so this needs to be big R, little r, and if we cross big R, little r with little r, we're going to get 50-50. So Mary is going to be big R, little r. And then for the one of Jack's parents, we have got 100% all dominant. So this one must be big R, which makes the offspring big R, and big R, little r. So now if we cross Mary and Jack, we know that they're both heterozygous, so this should tell you what they are going to give you the offspring of right away. So we'll do it in a Punnett square anyway, but we've got big R, little r, big R, little r. So that gives us big R, big R, big R, little r, big R, little r, and little r, little r. So we have 3 that are non-sufferers, and we've got 1 that is a sufferer of cystic fibrosis. So we have a 25% chance, so 1 out of 4 of the child having cystic fibrosis. We have 50% of the child being a carrier of cystic fibrosis, and we have 25% of them being the child being normal and not carrying or having the disease. So 75% of the child being a non-sufferer and 25% of the child having cystic fibrosis. So well done if you managed to get that correct. If not, go back and watch the family tree and pedigree charts again. So now we would do the starter pedigree worksheet in class, and you can find that in 14.3 starter pedigree worksheet in consumables. And you should pause the video and try to answer the worksheet before I go over the answers with you. So the answers are the oldest is 1 or 2. So because they're at the start. The youngest are 18 or 12 or 13 or 14 or 15. 6 and 7 are carriers of the disease, but their child, because they don't have the disease, but their child does have the disease. The youngest females are either 12, 14, 15 or 16. 10 and 11 are both affected by sickle cell anemia because they are grey. 2, 10, 15 and 16 are all the affected females. So 2, 10, 15, 16. 1, 5, 7, 8, 17 and 18 are all the unaffected males. So 1, 5, 7, 8, 17 and 18. The people that don't have children are 5, 12, 13, 14 and 15. And then the other question says add the girl and the husband of 14 to the chart. So that would be a husband because it's a square, and a girl would be a circle. And then 8 and 9 have 3 children would be the answer. So 1, 2, 3. So well done if you managed to get all of that worksheet correct. If you didn't, then make sure you understand where you've went wrong. Now you can complete the activities below to review your learning and make sure you understand variation and inheritance. So you can do the SQA PowerPoint that are found in one PowerPoint. You can do the past paper questions for key area questions that are found in the past papers for key area questions, and it would be the variation one. You can do the consumables, which are the fill in the keywords 15, and the review questions, which are 16 to 18 in the consumables. And then you can answer the questions on page 27 to 31 of the Multicellular Exam Practice book. And then you can do homework 4. We have now come to the end of the lesson. So you should now be able to write down and explain the definition of all of the genetic terms that were said at the start. You need to be able to successfully complete Punnett squares of monohybrid crosses, be able to predict phenotypes and genotypes from monohybrid crosses, explain why predicted and observed ratios are not always achieved, and describe how you are more likely to get a predicted ratio. Well done if you can confidently do all of that. If you can't, then you might need to watch the video again. And that is the end of this video tutorial. Woohoo! Thank you for listening.